So today I am going to talk about a bit of things that I do regularly, part of my research, but also would touch upon things that I don't do but I have interest in, and all of these things are going to be about fossils and in particular marine fossils. Okay. So I think I'll start with this picture, which is picture I think all of you know about. And the question is that except for the human character in this entire picture, what's common among them? What is it that all of them have in common? All of them are extinct, right? So that's something that's quite unique in terms of catching the attention of general public. You look at most of the Disney movies and often it characterizes the extinct organisms. So is it only this part that we are fascinated about, that there were organisms, animals which were there on the earth at some point of time and now they are gone? Is it the only motivation to know about them, or are there something else? So in order to understand exactly what is the importance of fossil organisms, we will do some very simple calculation. Okay? So I know that that's probably not the best thing to keep in a public lecture, that we are going to calculate something, but let me assure you it's going to be very, very simple. So what we are going to do is something called a diversity curve. What it does, it basically tracks geologic time and tries to put a number in terms of how many different types of species are there. And how do we know about it, or how do we create this? So what we try to do is to basically add numbers to these different time scales in terms of the number of species and try to figure out these very simple questions, such as we look at different kinds of species today. Was there a time when the number was more? Was there a time when the number was very low? Exactly how did it change over time? These are very simple questions. But in order to do that, we have to basically go through tons of literature where people have talked about that they found some fossils from a particular time, and how they know that particular time is through dating. I'm not going into the detail of it. At this point, we are simply saying that there is a way of knowing that age, and once you get that fossil, people are reporting it with their ages, where they are found from. And once you have this information, you can actually add stuff to this graph. And the way to add stuff is that if I have, let's say, 2 starfish fossils, I can add them to this time period called V, and this is somewhere around 600 million years ago. And then in the next part, we have 2 more fossils. These are also starfishes, and now it's slightly different because the colors indicate different types of species. Okay? And if I can keep on counting the color, it's going to tell me exactly how many different types of species are there in a particular time bin. Now it gets slightly more complicated because let's say this green one, this green species, it appeared in this V time, then we are not finding it in C time, but then we are again finding it in O time. Okay? And this is a continuous time. And we know that a species appears, it cannot just, disappear and then come back again. So that means this disappearance is basically we are not finding them, but it has to be there. Okay? As a result, what we do, we basically put them in between, and with a high confidence we say that the fact that they are absent in that time zone is simply because we are not finding them. Either they are not getting fossilized, or we just simply haven't looked enough. And all of these, once taken care of, you can start counting numbers. And I think we can actually start counting number right here, that this time v corresponds to 2 species, right? What about time c? I think now we can come up with the numbers that it's 3, right? Then in O it's 3 again. So we can keep on doing this and then add these specific points and then connect them. Voila, you get a diversity curve. So what this diversity curve tells you is exactly what is the number of species at a particular time interval and how it changes over time. And if we do this exercise for all kinds of species, we get something like this. Okay? Now this is interesting because the first question that I asked is, was the number of species very high or low back in time compared to today? This clearly shows that if you look at the 0 time, which is where we are right now, the points are very high up. So that means the total number of species that we see today is actually very high compared to many other times far back. Now let's ask this question, that exactly how many are there, or more importantly, how many were there, because that's the only way we can compare the number of species that we see today with all the number of species that have ever lived on the earth, which are represented by fossils. Okay? So this understanding, because we said that these curves are primarily made by these dots. Okay? So if you can count those dots, the total number of dots, that's going to tell you how many have ever lived over time. How do we count these dots under this curve? We basically try to calculate the area under the curve. Okay? Now this is a complicated curve, and the moment I try to say that area under the curve, it goes into the calculus, which becomes very complicated. Let's skip it. Let's make this curve very simple. So what is the best approximation of this curve? It's a, it's a triangle. So if I try this triangle, what it does, it basically, yes, there are some parts which are above, some parts which are below, but overall it's more like a triangle. And I'm going to calculate how much is there in that triangle, because that's also going to tell me that what is the total number of species that have ever lived on the earth, okay, represented by fossils. And then we are also going to count in comparison, where are we standing now? What percentage is this? So if we try to count this total species, I can come up with this kind of simple thing, that half the base times the height, it gives you an area of a triangle, and that also gives you something like the total number. Okay? But then we also say that this works very well if there is only one species counted once, but we have counted the same species multiple times. So we have to somehow normalize it, and one way of normalizing it is sort of divide it by the time a species generally lived. So the argument is somewhat like this. Let's say you are going to a college and you are trying to get an estimate that from, let's say, 1990 to 2000, how many students have been there in that college. You can go every year and count, but then you are going to count the same student maybe 4 times, because that same student first appeared in first year, second year, third year, and so on. So in order to correct that number, all you have to do is to get that number, but then divide it by 4 years, because that's the mean duration. And that's exactly what you can do for the fossils. You can basically counter that not all of them are unique, and there are some which are repeating multiple times, and in order to count those, I can divide it by this 5 million years. This 5 million is basically the number on an average a particular species lives. Okay? So even if you didn't really follow all the points, let's just do this calculation and see, in comparison to the modern day number of species, how many are we missing if we didn't study fossils. Okay? The number of fossils that are, or the number of species that are represented only by fossils comes out to be 98%. So essentially, all our understanding of biology, if we focus on biology of today, all the things that are living, we are going to get fantastic understanding of 2% of the total biology. Some people may think that that's too less. We are basically missing this 98% of all biota that you can get only from the fossils, and hence it's important to study them. It's not just that they are different, they are gone, they are also telling us a lot about how organisms evolve over a long time. That's something that's very hard to figure out in short time scale, which unfortunately humans cannot study things in human time scale, because our life cycle is finite, and we can, in the labs, we can look at things as they grow, as they change, only in that finite time scale. So the other aspect of it that we can find is from the fossils is this very long time scale and how groups evolve. So that's going to be the rest of the talk, I think, and one intuitive understanding of how tiny the present biodiversity is. If you look at this plot and just track the red line, that red line represents proportionally how much is the present day biodiversity. Look at how much is left in that triangle. That's all coming from the fossils. So again, to reiterate the point that if you skip the fossils in terms of our understanding of biology, we are going to be focusing on this 2%. And sometimes the understanding is very good about the 2%, but it's just very small. And hence, again, people have been interested in the fossils. Now, exactly what kind of things fossils offer? And this is the time where we are going to shift to my favorite group, which is the seashells. Why are we so fascinated about seashells? And this is something that I have been answering since my college days. What's so great about them? I mean, my friends used to tell me what's so great about them. Well, some of them taste good, others would look good, but is it enough? And most of the times my answer would be, no, they are fantastic because you see this and that. And as I grew older and over time, I never could finish this and that. They kept on adding. It's almost like you look at every avenue and you get something about the seashells, and they are very interesting and they give you a lot of information. So that's what we are going to look at next. So by the way, this is a famous quote by a paleontologist looking at that 98%, that to a first approximation, everything is dead, everything is extinct, because obviously 98% is a large number. And I think it's also paleontologist's favorite quote because it sort of justifies what we do. Going to the seashells, seashells actually comprise a large percentage of that 98%. Because they have calcium carbonate in their skeleton, they get preserved very well. And by seashell, I mean clams, snails, squids, octopuses are also seashells. It's only that they don't have the shell, they left it during their evolution at some point of time. But generally, seashells are all the things that you see when you take a walk on the beach. Some of them you can actually see on the land. Some of them actually became land dwellers later. But these are groups that we are talking about. And this is a general picture that you find, and all of them are telling you something different. Now, for a paleontologist, we are always interested in getting information from different parts of the world. And these different parts are not always necessarily geographically separated. It could be the same place, but different depths. And that's where these seashells are very handy, because they live in different parts of the sediments. So, some of them you are going to see right on the top of the sediments, which are always crawling and trying to go down. But then if you look deep, there are lots of seashells which are living inside these sediments, and some of them interact with the sea water directly, some of them are interacting with the sediments. So, each of them preserve unique environmental information which are interesting for past reconstruction of climate and environment. So, that is what we are going to see in some of the cases. Now, this is a famous person, it is just this picture is not very famous. This is young Darwin. The reason I am talking about young Darwin is he also started with this question that what makes group change? This is a vital question, and our understanding did not change much in terms of the major triggers. The major triggers remain the same. The groups change either because their environment, their biology around changes, or they change because the physical environment changes. So, either of these changes will make a group change, but then the question is how quickly does the group change? Does the group change in one generation, or does it take millions of years? And as it turns out, it can actually do both. Some of the changes can happen very quickly, even within generations, but sometimes it can take millions of years. And this connection that from quick change to long term change is not something that people understand very well. And therefore, if you can have a group which you can do experiments with because they are alive and also have fossil record, then it basically fits the entire spectrum. You can start with experiments and then go to really long term things. And that is something that I do and along with my students. So, we do look at things which are in the aquarium. So, that is that picture. And then in the aquarium setup, sometimes they do not behave the way they behave in natural system. So, that is why we also look at the natural system by going underwater observation, some scuba. Then whatever they are behaving in their natural setting, not all of them get preserved. So, we also need to know that once some of them get preserved, getting preserved means just getting the shells in a big shell hash where you are not finding all of them, but they are telling you something about the overall population. So, for that we need to understand that which are the ones that are never going to get preserved and which are the ones which are definitely going to get preserved. For that we do these kind of ecological sampling. What it means, we grab a lot of sediments and then look at the seashells, and because we know which are the alive ones, we can compare that from alive which are the ones which basically constitute these sediments just by with their shells. So, from live to dead, which are the ones which are going to make the transition, which are the ones which are going to swept away and never get preserved. Okay. So, these are some of the things that we use in terms of collecting the data. But then when it comes to long term studies or very deep time information, for that we go to field where things are getting preserved. So, this is a picture from Gujarat where seashells have been preserved, and some of the ages of these seashells are 20-30 million years. We also look at them, try to understand that in the long term how did they change. In other words, if we know the environmental change, can we connect it to the change of the seashells? With all of these data, eventually we try to make a model and then come up with a more quantitative understanding of how the world revolves around the seashells. So, just a few things that I am going to show you in the next few slides. First is how we understand in terms of ecological interaction from the fossils, where the biology or other biological organisms are playing a role. Then we will move on to how do you reconstruct climate using these seashells. So, this is again a major theme of ecology and any kind of change. Why do animals change? Because one of the major drivers of change is predation. All that means that always animals try to survive, and in order to survive they try to make changes in their body. So, if a seashell is very thin shelled, most likely it is going to be attacked by other organisms like crabs, and they are going to be crushed, and they are not going to pass on their genes to the next generation. So, natural selection will basically favor those groups which can change and make harder skeletons. But these questions are complicated because you can experimentally show it in smaller setting, but often when it go to the long term processes, it does not show that very well. And the other problem of this is, if you are trying to understand how predation works, like how a crab attacks, crabs are not going to get preserved, nor the crushed shells. Then how do you figure it out in the fossil record? So, you have to come up with some patterns which are good, and you can basically detect it in the record. So, I will come up with this kind of picture, which is fabulous for showing a predation event, where a fish is eating another fish. Although it looks fantastic, this is 1 in a million time. Mostly you are not going to get these kind of preservation. So, then do we just say that we cannot study it? Well, we look for things which are not so glamorous, so to speak, but it actually tells you more information, and some of these things are these. So, these things are always records of predation. For example, this particular hole basically tells you that it was attacked by a snail, it was eaten by a snail, where the snail extracted all the soft tissue from this snail, and it left behind this scar. And these scars are again telling you what kind of snail attacked it, how big was it, how quickly did it do it. All of these information eventually help us to make this model. The other example that I gave was this particular another snail, where you can see this curved line. This curved line was a result of an attack which did not kill it, but eventually regrow, and that is again something that you can put a number. In a population, how many such things are there? Okay. So, these are some of the examples that I am showing, and we basically study them in an aquarium like this, where we can all the features we can note down and show how it is relating to the size of the attacker, who is attacking it, how long it takes, and all these things, and eventually come up with a more quantitative model. And using these things, people have demonstrated that in that diversity curve, some of the major diversity changes have happened because of changes in the predation pattern. So, new predators came, your diversity shoots up, because all kinds of groups are trying to preserve themselves, all kinds of groups are trying to survive. As a result, they are picking and choosing different strategies. So, you identify predation in the fossil record, you can also identify what is the response to it. It tells you eventually why such diversity curves are developing, why there are wiggles, why there are sudden jumps. But it becomes even more complicated because there are sudden drops too. What are these drops? We know these are called extinction events, and often these extinction events cannot be related to simple biological changes. It is generally more environmentally dominated. Things like an asteroid impact, things like changes in the sea level composition, sea level itself, whether there is a glaciation, things like that. So, environmental change is also something that we really like to know. Can we relate changes in organisms with the environment? But again, it takes time. So let us try to look at another angle of this, where we are going to look at environments. What is this map? I hope everybody is sort of familiar with this map, and this is today's map, and you can see Mediterranean Sea in one side and Arabian Sea in the other side. Now, it would be really interesting if I tell you that there was a point of time not very far ago, it was only 20 million years ago, where if you wanted, you could have taken a boat from Arabian Sea and ended up going all the way to the Mediterranean Sea, because that land was not there. Now, the question is, today when we are talking about changes in the sea level, one of the major concern is that the sea level is fluctuating. There would be places where the sea configuration is going to change, and therefore people try to look at a model system where it has already been done and look at how animals responded to it. So therefore, this is one of the questions that people look at. So if you look at this map, this is a map where you see this common seaway. Okay? I'm just going to spend a little bit of time in terms of showing what is sea and what is land. So this is land. This is the part which is Africa, and this is the sea which connects the Mediterranean to all the way to Arabian Sea. This sea was called a Tethys Sea. And at some point of time, this got closed, making the modern day Arabian Sea and Mediterranean. Now the question is, if you just break it off, what happens to the animals which were once at one point of time connected? And that is where sort of our journey began. We started looking at fossils which we find from the western part of India, because they are providing this information. What happened before and after this closure? Did they change? Did they change because they were not connected to the previous times? So this is the place where we go to the field almost every year to collect more and more fossils, and to address this question, that exactly what happened once you close a seaway to the animals and also to the environment. And this is a typical picture from the field where one of my students is picking up a fossil, and even if it does not look anything like a sea, it was a sea. This was the sea floor, where nowadays if you go, every piece of the rock is fossiliferous. It has a lot of fossils. You have to use chisel and hammer to extract it. Now, we did this exercise for quite some time, and then eventually found a lot of seashells. It takes some time to identify them, and eventually we did that. Just to give you a sense of how it looks like in the field, it looks something like this. There are some places where you see the seashells on the ground where the sea bed is exposed, but at times it looks like layers, and they are showing you older to younger seashells and seabeds, and you can extract fossils from them. So, we use a combination of things, and we also use some dating techniques to exactly date each of these beds. So, this is how it looks like when you are not looking at fresh shells, but they are 20 million year old shells. It looks something like that, but our whole idea of this, or our main motivation of this, was to understand before and after how did they change. Now, I am going to show a bit of, again, some plots which might be difficult to understand. So, I am just going to give you start with a simple explanation. Let us say there are two regions and only one type of animal is there. We can characterize these two regions by what is the percentage of one animal is there, and you can compare them. You can do the same thing if there are two animals, because you are going to look at the percentage of two of these things, and then you can kind of think of it as a plot, and two places are getting plotted in that XY plot. If there are three animals, you can do the same thing again in a three dimensional plot. All it is telling you that if two regions are closer to each other in that plot, it means whatever animals they have, they also look similar. Similar type of species are there. Now, what happens if you have 70 such species? Then you are also plotting it in a 70 dimensional space. We cannot visualize it, but the math remains same, that the distance between the two locations in terms of their species composition is similar or shorter if they are sharing. If they are not sharing, they are very far apart. Okay? Now, if I use the same principle in this kind of plots, all it is telling you, if you figure out 2 points in this plot and they are close by, they have similar species composition. If you pick up a bucket of fossils from these two localities, similar type of seashells you are going to see. If they are very far apart in this plot, they are going to give you very different composition. Okay. Now, let us see what we are seeing here. So, before this seaway closure happened, you can see all these dots are sort of around the same place. There is no major gaps. You cross and you go to this other plot where it is after closure, you find that there is a cluster of these black dots, but they are very different from the rest of the materials. And if you now start to look at what are these rest of the materials are or the names are, you will find names like Italy, Germany, and the black dots, those are all from India. So, suddenly the Indian seashells became different from these things which are in Germany, Italy. But just like a few million years before, you see all of these black dots sitting next to Italy and Germany. So, basically the same seaway was transporting these seashells, keeping the same composition, but the moment you divide, you get very different composition. Well, it may sound very obvious, but it is something that today if you want to demonstrate, it becomes complicated. The other complicated stuff which is difficult to develop is what was the effect on the environment. You basically changed the circulation. There was a common circulation, you put a bridge. What happens to the circulation? Did it change? And that is something that we can figure out from the seashells again, but not what kind of seashells, rather what are their chemical composition telling us. Okay. So, I am going to now focus on this. So, a seashell is made up of calcium carbonate. It has couple of elements, calcium is one, carbon is one, oxygen is one. This oxygen has different kind of properties. We do not really think so much about it, we simply think it is a life saving gas, but it actually has couple of properties, and especially when it binds in different, depending on different properties. So, what kind of properties are we talking about? Oxygen is an element. It has certain proton numbers and also some neutron numbers. Now, depending on what kind of oxygen you are seeing, the neutron numbers can vary. These are called isotopes. Okay? And these isotopes are beautiful because if things are getting warmer or things are getting colder, these isotopic composition changes. But the problem is, a sea getting warmer 20 million years ago, you do not have the sea water to test whether the isotopic composition changed or to track. But you have the next best thing. The animals which were using the seawater H2O to make their shell CaCO3, that oxygen comes directly from the seawater. And therefore, if you can track these shells, it's going to tell you something about the seawater. And that's what we do. It's a bit complicated in the sense that you have to cut the shell, you have to see how they are growing, you have to take micro samples, take the powder, extract that oxygen to figure out whether these stable isotope ratios are changing or not. It can change because of two things. One is this temperature, the other one is salinity. Now, both of these things we played around, and there are instruments which can detect these ratios. Eventually, what we are trying to find, or what we found, was this is a curve which shows some stable isotope ratio change. Now, if you look at these two curves, the y-axis, there are some numbers written, these are ages. So, it shows that in some animals, these seashells, they are kind of going to the left and then they are kind of going to the right towards the top. If you look at the other one, it is also going towards the left, then going back to the right, but with different numbers. Okay, is not very informative at this point, unless we try to understand what these two seashells are. Now, if we put all of these things together, what it shows is a salinity. Okay? So, we looked at the temperature, but we are looking at tropics. So, temperature did not change, that is also we showed, but the salinity changed. And what the salinity shows is that it is actually decreasing over time. So, I will again explain what is a salinity. Salinity means in a bucket of water or in a glass of water, how much salt is there. So, if it is an ocean, you expect it to be salty. If it is not an ocean, if it is just rainwater, you do not expect it to be salty. Now, why do we find around this time things going from saltier to non-saltier? It is the same ocean. In fact, after you cut it off, it is a relatively limited area, and people expected that this would be a time when you expect for the water to become more saltier, but we are not finding it. So, what is it happening and why these two animals are behaving differently? So, that is why we need to know what is this animal. It is also a seashell, but it lives in the open ocean. That means does not matter what is happening in the ocean, it cannot live close to the shore. It lives in a reef or it lives in the middle of the ocean. So, what it is recording is in the middle of the ocean. So, its salinity is telling you kind of middle of the ocean signal. On the other hand, the other one, it is an oyster. Oysters do not live in the middle of the ocean. Oysters actually live in places which are next to the shore, and you have some puddle of water, sea water, but these puddle never goes back to the sea, and the oysters love to live there. Now, this helps us to solve the mystery. One mystery is why these animals are behaving differently. That mystery gets solved because one animal is living in the middle of the sea where the salinity is fantastic. The other animal is living in the coastal region where the water comes in. It is a saline water, water comes in, but it cannot go back. And when it stays there, it evaporates. Once it evaporates, the water goes up, not the salt. So, the salt increases, and these oysters basically live in that very, very salty water. Over time, they are getting the signature of extremely salty water and high salinity. The second question is, you see the difference between the two is decreasing. This is also something that kind of tells us that something is happening overall. It is not just coast, this particular open ocean, but the overall difference in salinity is decreasing. Today, if we compare it with today we find something very similar to the top, where the salinity difference is not so much, and this salinity difference kind of changes over time if you add a lot of fresh water. Today, I think this is an open question. Today, what is the major source in, let's say in India, of fresh water? Where do we get all our water from? Obviously from tube oils and taps and underground reservoirs, but where does it come from? Where do we get our drinking water from? Rivers. Where do the rivers get their water from? Rains, right? What is the biggest phenomena of rain in India? Monsoons. Monsoons were not there before. This is the time when we start finding monsoons. Why monsoons were not there before? Because the monsoons are things where the evaporated water with a lot of moisture bumps into a mountain, and then the mountain doesn't let it go anywhere, it climbs up, it condenses, then it rains, right? When it goes up, it gets colder, that is why it condenses. If you do not have that mountain, it cannot go like this, it cannot make this seasonal monsoon. And what is this mountain which created this? Why do we have monsoon? The next question is, which mountain is creating this monsoon, at least Indian monsoon? Himalayas. You remove Himalayas, you will not get monsoon. So this is kind of pointing that this is a time when Himalayas got to this height. Okay? What is the time we are talking about? We are talking about 24 million years, 23 million years, 17 million years. This is a time when Himalayas is actually getting very, very high. This is a time when these ocean moisture gets bumped into this rising mountain and starts this downpour of fresh water. This fresh water mixes with salt water, changes the composition. Before that, you do not see this compositional change. Okay? So, a tiny seashell growing up in the open ocean records this before and after. A tiny seashell which is sitting over there, things which are very, very high saline because of the evaporation is recording some of the differences. And when you put all of these two things together, you realize the development of monsoon, all from these seashells. You take out these simple tiny seashells from the history, we will enjoy monsoon, but we will never know what was the time like before monsoon. You remove them, we will never know how the seaways were before we put a bridge between Mediterranean and Arabian Sea. You remove them, you basically do not get an idea of how India looked like in broader sense without a Himalayas. So, these seashells are fantastic. I think I am pretty much convinced, and I think most of the people are close to being convinced that they are worth looking. Now, some of the things, one is that the present seashells, because they also have many of these properties that the old seashells have, you can do experiments with them and therefore change these things. So, for example, the stable isotopes and all these terms that I threw, all of these are done first on living seashells and changing the environment, changing the salinity and seeing exactly how do they respond. So, that is the biggest advantage. But the problem of this is you can only look at the experiments in a short time scale. It gives you fantastic resolution, no question about that, but only very short time scale. The next question is when you can look at these things into a slightly larger time scale, maybe 1000 years, then you get a more realistic understanding, but you lose the resolution because some of the things are not going to get preserved. Finally, you go to the fossils, you get the best temporal scale, but you can never be certain that maybe some of them got preserved because they had thicker shell, or they were living in a place where they were getting preserved very well. So, it is finally a combined approach that often makes the best of the scenario, and that is why I think the life of a paleontologist is also very interesting. We are never in one place, we are always looking for different approaches, and we get to do scuba, we get to do experiments, we get to do fossil hunting all in one. I am very grateful to Seashells and all other members of my lab, past and present, for giving me the wonderful and exciting life. Thank you.